am Madam Joyce from Educare TZ. I'm teaching biology, and today I'll be with you to discuss our topic, which is gaseous exchange and respiration. And here we are going to deal with our subtopic, which is respiration. So my student, in our topic, which is respiration, we have seen that we have got two types of respiration, which is aerobic and anaerobic respiration. So in aerobic respiration, it involves just mainly three stages. That is the first stage, which is glycolysis. The second stage, it is Krebs cycle, and the third stage, it is oxidative phosphorylation. So hizo ndizo stage ambazo zina involved kwenye aerobic respiration. But looking on the glycolysis, we have already covered about it on the previous session. And in this session, my student, we are going to start discussing the Krebs cycle, which is the second stage of aerobic respiration. So now let's see what is it. The second stage of aerobic respiration, as I have said, it is just a Krebs cycle. So what is that Krebs cycle? So simply here we can say that Krebs cycle is also known as citric acid cycle, au kwa jina lingine tunaita tricarboxylic acid cycle, TCA. So ukikutana na hayo majina usichanganyikiwe. Hapo tujue kwamba umeambiwa uelezee kuhusu hiyo Krebs cycle. That is the citric acid cycle. So hapo it can be abbreviated as CAC, that is citric acid cycle, or TCA, tricarboxylic acid cycle. So when oxygen is available, the pyruvate produced during glycolysis enters the Krebs cycle, named after Sir Hans Adolf Krebs in 1800 and 1881, who worked out the details of the cycle in 1830. So Krebs cycle takes place in the matrix of the mitochondria. So the word Krebs, that is Krebs cycle. So this word Krebs, it comes from this scientist ambaye anaitwa Sir Hans Adolf Krebs. Huyu ndiye ambaye alifanya utafiti kuhusu the details of the cycle. Alifanya in that year 1930. So ndio maana akaita hii cycle as Krebs cycle. So the word Krebs, it comes from his name. That is the Sir Hans Adolf Krebs. So and asema kwamba hii Krebs cycle always takes place in the matrix of the mitochondria. We have seen in the previous session just introduction. That is we were discussing on the transition stage between the glycolysis and the between the glycolysis and the Krebs cycle. Before Krebs cycle pale tuliona kuna formation ya acetyl coenzyme A and so many things. So na obvious tumesema kwamba hii Krebs cycle, remember, it is just taking place within the mitochondria. Lakini ile first stage of aerobic respiration ambayo ni glycolysis, tulisema ina takes place within the cytoplasm of a cell and not within the mitochondria. So in this stage now let's discuss on the steps involved in the Krebs cycle. Steps which are involved in the Krebs cycle. So the Krebs cycle consists of the series of the enzyme catalyzed reaction. So involves the following steps. So kwenye kila reaction ambayo hipo ndani ya Krebs cycle pia ina be facilitated, au tunaweza tukasema kwamba it is controlled with the enzymes. So enzyme ndio ambaye ata control the different kinds of reactions which are normally taking place within the cell or within the matrix of a mitochondria. And now let's start looking on the first stage of the Krebs cycle, and this is, we can say that the first stage in the reaction of this acetyl coenzyme A with the oxaloacetate to form citrate, in which the acetyl coenzyme A, which is 2-carbon compound, is joined to oxaloacetate, which is 4-carbon compound, to form a citrate, which is a 6-carbon compound. Remember, before we just started looking on the Krebs cycle, we're discussing on the transition state. And on the transition state, we just say that we convert that pyruvic acid in order to form the acetyl coenzyme A. So hile acetyl coenzyme A ndio ambayo sasa itaendelea na process ya Krebs cycle. Na hapa kwenye hii first stage ya Krebs cycle tumeona kwamba inahusisha ile acetyl coenzyme A ambayo ilitokea kwenye pyruvic acid au pyruvate. So ile acetyl coenzyme A tumesema kwamba itaungana na oxaloacetate kuform citrate. Hii acetyl coenzyme A ni 2-carbon compound. So itaungana na oxaloacetate ambayo ni 4-carbon compound to form citrate ambayo ni 6-carbon compound. And this is the reaction which is taking place. So this process will require the input of water, and it is characterized by a citrate synthetase enzyme. So now you can see our reactions. Notice we have this one, which is acetyl coenzyme A akareacting na huyo oxaloacetate, and here I have said that this reaction involves the input of water. So we need the presence of water so that it can now function this reaction. So inaweza ikasaidia hii reaction to take place. So pia tumesema hii reaction it is controlled by enzyme, ambaye enzyme anahusika control hiyo reaction anaitwa citrate synthase. So after that, after the joining of the acetyl coenzyme A and the oxaloacetate, it will now form the citrate together with the coenzyme A and other product. So our main product that we need for the formation of ATP, it is citrate. So citrate ndio ambayo tutaenda kumove now kwenye the second stage. Zile nyingine zitakuwa hazina function kwenye our Krebs cycle. So it is better to know those products that are formed after this reaction. So now let's discuss on the second stage of a glycolysis, of a Krebs cycle. And the second stage, which is the second stage, is the formation of isocitrate. The formation of isocitrate. So in the presence of aconitase enzyme, citrate is converted into isocitrate. So as I have said, pale kwenye hile baada ya kutengeneza ile 6-carbon compound ambayo ni citrate, ndio ambayo tutaichukua tutaenda nayo kwenye the second stage of a Krebs cycle. Ambapo sasa kwenye hii second stage of a Krebs cycle tumesema kwamba inahusika kutengeneza hii isocitrate. Je, hiyo isocitrate inatengenezwaje? Tumesema kwamba hapa in the presence of the aconitase enzyme, ile citrate is converted into isocitrate. Kwa hii citrate hapa itabi converted, itabadilishwa kwenda kupata isocitrate. And that reaction, it is controlled by this enzyme, which is the aconitase enzyme. And this process is accomplished by dehydrogenation and rehydrogenation to yield an isomer called the isocitrate. So in order to convert ile citrate mpaka tuje tupate isocitrate, tunasema kwamba it involves dehydrogenation, meaning that there is a removal of water from that reaction. And apart from removing of water, also it involves the rehydrogenation, meaning that we tend to add water again. Na ndio maana tutaenda kuangalia kwenye reaction yake tutaona. So that inaweza kupata hii isomer ya isocitrate. Kwa ina maana ile citrate, hii isocitrate is just an isomer of a citrate. So this one is the reaction. So unaweza ukaona hii ni structure ya citrate, tumesema kwamba hii reaction ina involves dehydrogenation of water. So tuki-involves dehydrogenation of water under this enzyme ambayo inaitwa aconitase, tutapata a transition state or transitional product ambayo inaitwa cis aconitase. So we don't need this cis aconitase, aconitate. We need the presence of isocitrate. So how we can form isocitrate? Here simply we can say that we tend to add again water. So after addition of water now is whereby we can form the isocitrate. Now you can see the difference which is occurring between the isocitrate, the aconitase, cis aconitase, aconitate, and the citrate. So unaweza ukaona all of them, they have 5 carbon compound, and here you can see this presence of 5 carbon compound, but after addition of water now you can see this addition of water here, hydrogen atom and hydroxy atom, so that it can be from the isocitrate molecule. So unaweza ukaona kwamba kwenye kila reaction tunahitaji uwepo wa enzyme. So huwezi ukaelezea reaction ambayo inapatikana kwenye Krebs cycle bila kuweka uwepo wa enzyme. So that is the first stage, the second stage of glycolysis, which is occurring in the matrix. So my students, for more clarification on the other stages of Krebs cycle, I would like to invite you into the next part of this session, so that we can have now all of the steps of the Krebs cycle. So thank you for your time and for listening.